Dear guests, welcome to the online launch event of Mindray ToRCH panel. As we know, ToRCH detection is closely related to the health of pregnant women and neonates. We are so excited to launch the ToRCH panel, a result of our consistent R&D efforts. With this panel, we can provide high-quality assays to meet different clinical needs. I'm gonna show you the ToRCH development process and their performance. We also have the contents of clinical applications and scenarios of ToRCH tests. Now, let's move on to our first stop, Mindray R&D Center, and take a close look at the development journey.
In vitro diagnostic is a bridge that leads science and technology to clinical application, especially for chemiluminescent immunoassay. For CLIA, accuracy is the most critical feature in assay performance. By building up a closed-loop system that encompasses raw materials, such as antibodies, antigens, research, also development process, and matched instrumentation and reagent formulation, Mindray ensures each testing accurate for our customers. Currently, we have 4 R&D centers for IVD immunoassay, which are located across the world, and our R&D investment accounts for 10% of Mindray's revenue. In the near future, Mindray R&D team will expand and grow at a faster pace, along with continuous R&D investment. In order to better develop reagent for immunoassay, we have acquired HiTest to bring together a wider group of scientists to unlock more possibilities, which with immunodiagnostics, HiTest as a world-renowned IVD raw material supplier has been committed to developing innovative products that combine industry, academic, and hospital-based people. HiTest has developed a series of high-quality monoclonal antibodies products for cardiac biomarkers, for clinical diagnostic and prognostic applications, providing more valuable evidence for the companion diagnostics of cardiac diseases. Besides cardiac specialty, HiTest also provides high-quality antibodies and antigens using innovative and advanced technologies in infectious diseases, inflammation, and many others. With a keen focus on scientific research, HiTest designs and creates materials for standardization and harmonization of clinical testing. HiTest has won the NACB Distinguished Abstracts Award for its AACC posters in 2008, 2011, and 2016, and has published more than 50 papers in peer-reviewed journals, making significant contributions to the research on biomarkers. Mindray will be more dedicated to innovating technologies and methods to design and develop assays with increasing quality and quantity for our immunoassay analyzers, providing additional values to our customers.
From 2019 to 2022, more than 26 engineers and scientists were actually involved with the development of the ToRCH projects to meet our customers' core requirements. Our everyday work has been focusing on carefully carrying out the product design, studying the raw material processing, and improving the performance of our ToRCH reagents. Based on Mindray's MPI innovation system and advanced technical platform, hundreds of process parameters were optimized and more than 200,000 test results were evaluated for each assay in ToRCH projects. We finally developed highly sensitive and specific ToRCH assays, providing accurate and efficient diagnostic results for our customers.
Dr. He has shown us the R&D capacity of Mindray clear and told the story about HiTest. And Dr. Chen gave up a brief introduction to the R&D journey of Mindray ToRCH panel. Now you must be curious about how it performs. Now let's invite Dr. Li to give more details on this.
Hello everyone, I'm Dr. Li Ke, Director of Managing IVD R&D. Today I will introduce the performance of Mindray ToRCH panel. My speech consists of 3 parts. First, I'll introduce the clinical value of ToRCH and its key requirements for the reagents. Second, I'll discuss challenges clinical laboratory facing and how to address them. Finally, I'll showcase the excellent performance of Mindray ToRCH assay. ToRCH is the acronym for Toxoplasmosis, other agents, rubella, cytomegalovirus, and herpes simplex virus. Pregnant women infected with ToRCH often have mild or no symptoms. The virus can be transmitted to the fetus through the placenta. The table at the bottom shows the rate of congenital or neonate infection caused by ToRCH pathogens. And we can see that the birth defects caused by ToRCH infection accounted for 7% to 10% of all birth defects. As shown in the figure in upper left, the fetus infection rate increases with the increase of gestational age. Infection in the first trimester of pregnancy can cause serious damage to the fetus. So, pregnant women should be examined as early as possible. As shown in the table in the upper right, primary infection can cause a higher fetal infection rate and more serious fetal injury than recurrent infection. It's very important to distinguish between primary infection and recurrent infection so as to better guide clinicians in making medical decisions. The combined use of serology and PCR provides a helpful method to maximize the diagnosis rate of common virus and toxoplasmosis infections. Thorough screening is often the first step to take. Its value is to determine the immune status and to screen high-risk patients, which helps to guide further diagnosis and to take preventive measures. PCR is commonly used as a confirmatory test of acute infection to determine whether the fetus is infected. In addition, PCR examination is invasive. By delving deep into national guidance from regions such as Canada, the USA, and Europe, and the diagnosis and treatment process, we found that IgG-IgM combined testing and continuous monitoring can effectively distinguish among primary, past, and recurrent infections and provide more explanation for clinical diagnosis. We have also explored the key benefits of serological detection. For IgG test, its benefits include quantitative detection, high clinical sensitivity and specificity, a wide detection range, and good reproducibility when used for IgM tests. It provides high clinical sensitivity and specificity. It also ensures high clinical specificity for IgG avidity test. Two major challenges remain to be solved for serological ToRCH test in the laboratory. The first is the large differences in the test results between different systems. As shown in the table at the bottom, the maximum value is 6.7 and the minimum is 0.96. So the difference is large. The reason for this includes different raw materials, different traceability standards, different reaction models, and the absence of golden standards. The second challenge is how to balance the IgM sensitivity and specificity. The prevalence rate varies among different regions as shown in the figures. The false positive ratio is affected by the prevalence rate. The lower the prevalence rate, the higher the false positive ratio. Since the seroprevalence of ToRCH IgM is very low in most regions, false positives are very common. Besides the prevalence rate, the false positive ratio is also affected by other facts, such as pathogen cross-reaction, autoantibody interference, and polyclonal stimulation. In addition, IgM antibodies may persist for months to years in some individuals after acute infection. It's very hard to distinguish between primary and non-primary infection. As mentioned earlier, high sensitivity and specificity matters a lot to the clinical performance of reagents. To achieve that end, Mindray has been working consistently to improve the raw materials, process, formulas, and reaction models. According to the project characteristics and process requirements, we spend much more of our time on the design and development of immunodominant proteins and immunodominant epitopes. Good raw materials are fundamental to high sensitivity and specificity. Mature, stable, and innovative technology is another guarantee of satisfactory performance. For the coating and labeling process, we choose the particles and ALP that can best meet the assay requirements. To improve the sensitivity and specificity, we spend a lot of time thoroughly studying the parameters using DOE specific means. With a robust design, the boundary of each parameter was determined to ensure the reproducibility of the process. At the formulation level, we investigated the interference mechanism and effectively classified different types of interference to help enhance the anti-interference capacity. Hundreds of commercial and uniquely designed blockers were studied and applied to reduce 4 types of interference. For the IgM assay, we designed a capture method to reduce the IgG and RF interference. The 3 quantitative kits are fabricated with materials with international standard units and the latest standardized traceability. ToRCH IgG includes specific IgG and fetus DT test standardization. CMV IgG adapts the first international standard and features long-term stability. RV IgG comes with a cutoff value of 10 IU per milliliter as recommended by WHO. In addition, Mindray ToRCH IgG assay provides a wide detection range and a 10-time auto-dilution ratio to expand the reportable range, making the detection of increased IgG levels earlier. Mindray ToRCH IgG assay requires diluting a smaller proportion of samples compared to mainstream competing products. With the improved raw materials, process, formulas, reaction models, and the standardized traceability, Mindray ToRCH reagents deliver excellent seroconversion sensitivity. Mindray ToRCH has a good detection rate for samples that are positive in multiple systems, or for true positive samples such as seroconversion panels. The combined examination by Mindray ToRCH, IgG, and IgM can accurately disclose previous infection and promptly detect ToRCH infections not ruled out previously. In order to confirm the sensitivity and the specificity of Mindray ToRCH kits, we conducted tests in over 5 clinical bases, both in and outside China. Although the positive rate varies among different countries and regions, the kits found to have great specificity for ToRCH IgG and ToRCH IgM, and excellent sensitivity for RV and CMV IgG. And there are no significant differences between the Chinese and the international basis from the perspective of the wide coverage of the clinical tests. The Mindray ToRCH kits support diverse sample types with multiple quality controls and low sample volumes, which ensure great ease and convenience during clinical detection. By realizing the clinical value of ToRCH and the core requirements for the reagents, Mindray has solved the challenges facing clinical laboratory and made Mindray ToRCH assays accessibly in clinical application.
Now you have learned the excellent performance of Mindray ToRCH panel and how our R&D team has worked to ensure the quality of our products. This is internal evidence. We are also conducting external evaluations globally for further validation. Today, let's take a look at one of our evaluation sites, Colombia. Please follow me and see what happens there.
Como sabemos, en alianza con Mindray y ANAR, hace varios meses se realizó la verificación del panel ToRCH específicamente para toxoplasma, rubeola, citomegalovirus, tanto para IgG como para IgM. Doctora Ana, cuénteme qué tan importante es para ANAR contar con el portafolio del panel ToRCH en su catálogo.
Para nosotros es muy importante debido a la alta demanda que hemos tenido de los laboratorios y de nuestros clientes. Esta es una prueba de rutina y además porque atiende población muy especial como lo son maternas y neonatos. Es una prueba que se procesa en la mayoría de laboratorios del país.
Súper interesante, doctora, qué bueno. Doctora Mónica, debido a que la doctora Mónica Martínez fue la persona que apoyó en la verificación de este panel ToRCH en las instalaciones de ANAR Health Technologies. Doctora, cuéntenos cómo fue su experiencia y cómo fueron los resultados que se obtuvieron.
Se realizó un estudio de precisión siguiendo los lineamientos de la guía del CLCI EP15A3 con un proceso de repetibilidad y precisión intermedia durante 5 días con corridas de 5 réplicas de cada nivel de control por día, donde lo que se buscaba era evaluar que no existiera error aleatorio significativo que impactara clínicamente los resultados de los pacientes. Con los resultados obtenidos en este estudio de verificación, se evidenció que la variación intradía e interdía cumplió con los coeficientes de variación y desviaciones estándares declarados por manufactura y los requisitos de calidad establecidos siguiendo así el algoritmo de la guía. En el estudio de verificación se realizó igualmente una comparación de métodos donde se obtuvo un porcentaje de concordancia mayor al 80% para todas las pruebas, cumpliendo así el criterio de aceptación.
Doctora Mónica, muchas gracias por el resumen de la evaluación que hizo y por el análisis. Doctora Ana, de acuerdo a estos resultados que la doctora Mónica nos brinda, que estamos seguros que vamos a poder comenzar a trabajar con todos nuestros clientes potenciales.
Claro que sí, estamos seguros de eso y con el apoyo que Mindray nos brinda y haremos un trabajo en conjunto, obviamente en beneficio de nuestros clientes, de los pacientes y por supuesto de las dos compañías, Mindray y ANAR.
Al realizar el análisis del estudio de verificación de la precisión y la comparación de métodos para ToRCH, se confirma que el procedimiento analítico es adecuado para el uso en los diferentes laboratorios clínicos, ya que la imprecisión, la veracidad y la incertidumbre de medición no son estadísticamente ni clínicamente significativos para que puedan interferir con los resultados de los pacientes.
Como vimos, son excelentes resultados. Es un panel que es apto para el diagnóstico clínico. Doctoras, muchísimas gracias.
A ustedes, gracias por la invitación, sobre todo gracias por el apoyo y el soporte que nos brindan a nosotros como sus aliados.
As mentioned earlier, Colombia is just one of our many external evaluation sites across the world. Here, we have our clinical expert, Dr. Tina, to show us more details of our external evaluation results.
Mindray conducted the clinical verification of the 6 parameters in ToRCH panel at 2 reference sites in Colombia between October 2021 and May 2022. The comparative instruments we used were the 2 well-known competitive brands in the market. Mindray Toxo IgG, CMV IgG, and Rubella IgG were found to have satisfactory total coincidence rates with the 2 comparative instruments. The total coincidence rates of Rubella IgG with the 2 comparative instruments were 86% and 88% respectively. The percentages were 92% and 91% respectively for Toxo IgG, 98% and 94% respectively for CMV IgG. It is worth mentioning that the kappa incidences of these 3 parameters compared with these 2 comparative instruments were all greater than 0.8, indicating that the agreement of the comparison results is close to perfect. In addition, the total coincidence rates of Mindray Toxo IgM, CMV IgM, and Rubella IgM compared with one of the comparative instruments were 88%, 93%, and 92% respectively. Moreover, the total coincidence rates of the 3 parameters with the other comparative instruments were all around 80%, showing good clinical performances. Mindray also carried out a clinical evaluation study with a large sample size in 4 large hospitals and chain laboratories in China. The study involved using 3 different mainstream brands instruments in the market and 1 common ELISA method. The results showed that the positive and negative coincidence rates of Mindray's ToRCH panel were similar to those of the comparative instruments and methods. In addition to the evaluation mentioned earlier, we are also going to evaluate the 6 parameters of ToRCH panel in Ukraine, Italy, Colombia, and other countries. More and more international evaluation requirements have been added to the evaluation list of Mandarin ToRCH, which is attributed to both the growing demand of ToRCH tests from our international customers and the Mandarin huge KOL pool. At present, we have cooperated with local KOLs and customers in the international evaluation centers in Europe, Asia Pacific, Middle East, Latin America, and other countries and regions, which will help a lot in ensuring the accuracy and the Mandarin IVD product for different populations in different parts of the world.
Although ToRCH is a routine clear panel, we thought we would like to take this chance to reveal its value in different clinical settings. It is widely used in obstetrics and neonatology departments. Today, we are honored to have Professor Sergio Bernardini, Chair of the Emerging Technologies Division of IFCC, here with us. In his presentation, Professor Sergio is going to update the information about the clinical applications of ToRCH tests.
Dear colleagues, hello everybody, and thanks to Mindray for the kind invitation to give a talk about the ToRCH complex. We are going to answer to the question, why testing for ToRCH complex during pregnancy and perinatal age? And I'd like to introduce you the UNICEF For Every Child project, which has the goal of ending preventable newborn death and stillbirth by 2030. You know that half of all under 5 deaths still occur in the first months of life, and 2 million stillbirths occur every year. Times of health crisis, such as the COVID-19 pandemic, reveal the weakness of health system and threaten to turn back a decade of hard-won progress. The Every Newborn Action Plan provides countries with a roadmap for ending preventable newborn death and stillbirth and reducing disability by 2030. And the main actions are that every pregnant woman has 4 or more antenatal care contacts, including laboratory tests. Every birth is attended by skilled health personnel. Every woman and newborn receives early routine postnatal care within 2 days. Every small and every sick newborn receives care. Also, the WHO delivered a guideline on antenatal care in 2016, and you can find 49 recommendations grouped into 5 topic areas: nutritional interventions, maternal and fetal assessments, including laboratory tests, preventive measures, intervention for common physiological symptoms, health system interventions to improve the utilization quality of antenatal care. And here you can see that inside maternal assessment, there is in particular the screening for infectious disease, in particular HIV, syphilis, and tuberculosis. So, what about the ToRCH complex? ToRCH complex included toxoplasmosis, others, and in particular syphilis, hepatitis B, HIV, parvovirus B19, varicella zoster, Coxsackie, listeria, and recently Zika virus. And then rubella, cytomegalovirus, herpes simplex 1 and 2. And on the right side of the slide, you can see how many organ damage can be determined by the ToRCH complex members, and that can lead to newborn death and to important clinical sequelae later in life. Obviously, there are some maternal risk factors for ToRCH infections, in particular, lapsed immunizations, immunodepression in HIV, tumors, transplant, transient immunodepressions, sexually transmitted infections, animal exposure during pregnancy. The timing of transmission and the route of transmission can be different for different pathogens. Prenatally, by the transplacental route, perinatally, by blood or vaginal secretions, via sexual contact to a susceptible mother, HIV, hepatitis B, and syphilis in particular, with the exception of HSV infection during the first trimester have the worst outcome. Initial evidence of infection may be seen during the intrauterine period, at birth, in infancy, or not even until years later. And the maternal history is a key area of investigation for patients who have a concern for congenital infections. The history of a febrile illness with or without rashes and poor maternal weight gain could raise concern for clinicians. Furthermore, fetal abnormalities such as intracranial calcifications may be detected in routine or specific maternal testing may occur. Fetal loss can occur, particularly with infections during the first trimester. But just look at toxoplasmosis, which is determined by the Toxoplasma gondii, which is a typical endocellular coccid protozoan belonging to the phylum Sporozoa. The final hosts are the domestic cat and other felines. The parasite multiplies in the intestinal cell of the cat, while in the intermediate hosts, the reproductive cycle is extraintestinal with passage in the bloodstream. Oocysts are released with cat feces into the external environment and ingested by various animals, causing acute and chronic infection of various tissues. Then, at first, the oocytes are captured by macrophages and make the so-called tachyzoids. Then the toxoplasma are encysted in the tissues with consequent chronicization of the infection in the so-called brachyzoids stage. Toxoplasma gondii oocyst transmission occurs by ingesting the infected tissue or inhaling the fecal particles. Transplacental transmission causes congenital toxoplasmosis. This is most commonly occur in the third trimester of pregnancy. However, earlier the infection, more severe will be the congenital malformation. Infection by toxoplasmosis is ubiquitous, but susceptibility to infection varies between countries depending from the seroprevalence in pregnant women, which is different. In US, 15%. In Great Britain, it's been reported from 10 to 18%. In Spain, from 19 to 29%. In France and Greece, 55%. And in Italy, 40%, which means that the susceptible pregnant women are 60%. Maternal-fetal transmission is possible in case of a first infection in progress pregnancy, in a woman therefore previously seronegative. And the global risk of transmission to the fetus is between 18 and 44%, but it depends on the gestational age, varying between 6 and 26% in case of maternal infection at 7-15 weeks of amenorrhea, and between 32 and 93% between 29 and 34 weeks of amenorrhea. The global percentage of clinical manifestations among infants from infected mothers vary between 14 and 27%. The primary manifestation of congenital toxoplasmosis include intrauterine growth retardation and low birth weight, hepatosplenomegaly, jaundice, chorioretinitis, intraparenchymal calcification, and anemia. Less commonly, petechiae, hydrocephalus, and microcephaly. In particular, a newborn with a concern for congenital toxoplasmosis, evaluation should include laboratory testing, consultation, in particular by an ophthalmologist, and radiological studies. The diagnosis of acute form is possible in the mother with the use of serial serological assay that show an increase in the IgG and IgM antibody titer. In fact, the asymptomatic infection is very common, and IgM can persist for over 12 months, and even IgG titers can be elevated for years, leading to false positive for an acute infection. This is why it's important a serial serological assay. In immunosuppressed people with tumor, transplant, HIV, and so on, it's often a reactivation, the reactivation of an asymptomatic latent infection. If the IgG and IgM antibodies are negative and there is a clinical suspect, to rule out an active infection, molecular studies can be carried out. And prenatal diagnosis uses molecular testing amniotic fluid, which can also be used to search for trophozoites and cystis. Neonatal diagnosis is confirmed by the presence of maternal IgG and IgM in the newborn's blood. A prenatal screening is offered in Italy, France, Switzerland, Austria, Germany. It consists in a first serological test to establish the immunological status and or detect early disease in pregnancy with specific IgG and IgM antitoxoplasma antibodies. In case of seronegativity, and therefore of susceptibility to infection, the test is repeated regularly, monthly or quarterly until the end of pregnancy, given the few or no symptoms of the disease. In other countries, in particular in Poland, Denmark, Sweden, Brazil, and some areas of the United States, instead of the prenatal screening, the newborn screening is done, which search for IgM on blood collected with Guthrie card at birth to treat with drugs the newborn independently from the symptoms. But this type of approach is ineffective on irreversible damage already present at the birth. Then it should be adopted if other option, and in particular prenatal screening, is not feasible. Start to move to others. The first is safeness. Infection during pregnancy can cause serious fetal damage due to the possibility of vertical transmission, but transplacental and at the time of delivery. And this can lead to intrauterine fetal death up to 1/3 of cases, preterm birth, congenital anomalies, active infection, neonatal infection. And primary and secondary syphilis is if left untreated, result a risk of fetal transmission of 70 to 100%. If treated, just 1.8%. So you can understand how prevention and diagnosis of congenital syphilis depends on the diagnosis and treatment of infection in pregnant women, and the screening is strongly recommended. The screening can be accomplished with non-specific tests for treponema, as for instance the Venereal Diseases Research Laboratories, but in particular with specific tests for treponema. Treponema pallidum agglutination assay, treponema pallidum particle agglutination assays, fluorescent treponemal antibody absorbance test, and treponemal IgG-IgM determined by chemiluminescent assay or immunoenzymatic assay. Specific tests for treponema generally remain positive throughout the life, regardless of the therapy and activity of the disease. And here you can see the incidence or new cases of syphilis in 2020, 7.1 million. And here you can see the line which was expected in the project of, on the WHO project started in 2016 to that expect almost the elimination of new infection by 2030. In this report, 2021 report, you can see that we are far from the point in 2020 is very far from the line which was expected. So the serological screening for syphilis carried out with specific tests for treponema is strongly recommended in all women at the first visit and at the end of the pregnancy at least. Hepatitis B is another member of the others and is also included in the project of WHO that I told you before. Here you can see the expected line toward the almost zero cases in 2030 and the number of infection in 2019. And in this case, the result is quite good, but we are still far from the final target, the final goal. In any case, the research of the surface antigen is universally recommended in pregnancy in order to prevent mother-to-child transmission. The execution in the first trimester allows for early diagnosis, the execution of further confirmatory examination, for example, antigen E, vaccination of pregnant women at risk, and the identification of children to be subjected to immunoprophylaxis and postnatal vaccination. In North American, recommendations are to perform the exam in the first trimester and repeat it in women at risk in advanced stage or at the childbirth. HIV is also a member of others, and also HB is included in the WHO project that I told you before for syphilis and for HEB. And you can see then that the results in 2019 were quite good, but quite good, but still still far from the final target. The early identification of HIV positive women during pregnancy is aimed at reducing the risk of maternal fetal transmission of the virus too. In the absence of any preventive intervention, the transmission is elevated 15 to 30%. Over 80% of the transmission of the virus occur in the third trimester of pregnancy during childbirth. So by implementing preventive intervention, cesarean delivery scheduled at 38 weeks of gestational age, and in any case before the rupture of membranes, zidovudine prophylaxis and infant artificial feeding, the risk of transmission is reduced to less than 1%. So you can imagine how relevant is the screening of HIV. And then rubella. Serological screening for rubella susceptibility should be offered to all women at the beginning of pregnancy. In women found to be susceptible to rubella, the test should be repeated at 17 weeks and vaccination should be scheduled in the postnatal period in order to protect any future pregnancies. The so-called congenital triad defect in rubella are microcephaly, patent ductus arteriosus, and cataracts. But there should be also low birth weight, hepatosplenomegaly, ventricular septal defect, petechial rash, and congenital sensory neural hearing loss. This is why immunoassay such enzyme immunoassay, chemiluminescence assay, radial hemolysis are valid for the screening of anti-rubella antibodies and for identification of susceptible women. Then cytomegalovirus. Cytomegalovirus infection is relatively common among women in reproductive age, with a seroprevalence between 45 and 100%, which usually is higher in South America, Africa, and Asia than in Western Europe and US. The disease is strictly species-specific, and humans are the only reservoir. Transmission can take place horizontally by direct or indirect person-to-person contact, or vertically during pregnancy. The virus is found in urine, in the oropharyngeal secretion, in the cervical or vaginal secretion, in semen, milk, tears, and blood. The diffusion of infections requires close and prolonged contact with the infected patient and object. With primary maternal cytomegalovirus infection, the overall risk of transmission to the fetus is approximately 30 to 40%. And although vertical transmission may occur at any stage of pregnancy, the risk of transmission is greatest in the third trimester. Transmission rate, indeed, the transmission rate for primary infection are 30% in the first trimester, 34–38% in the second trimester, and 40 to 72% in the third trimester. More serious fetal sequelae occur after maternal infection during the first trimester. Of those fetus infected in utero, after a primary infection, 12 to 18% will have signs and symptoms of cytomegalovirus infection at the birth, and up to 25% will develop sequelae. Approximately 30% of severely infected infants die, and 60 to 80% of survivors have severe neurologic morbidity. Congenital cytomegalovirus may be suspected prenatally after a documented maternal primary infection, or more commonly, because universal screening is still not recommended, after ultrasound, finding suggestion for infection, in particular calcification, and that you can see in the right side of the slide, intracranial calcifications, but also microcephaly, hydrops fetalis, growth or retardation, hepatosplenomegaly, and so on. So although adults with primary cytomegalovirus infection are usually asymptomatic, individuals may experience a mononucleosis-like syndrome that should be noted in pregnant women with fever, chills, myalgias, malaise, leukocytosis, lymphocytosis, abnormal liver function tests, and lymphadenopathy. After the primary infection, cytomegalovirus remains latent in host cells. A recurrent or secondary infection can occur. Secondary infection with intermittent viral excretion in the presence of host immunity depression can occur after reactivation of the latent endogenous cytomegalovirus strain, or by a reinfection with a different exogenous viral strain. Cytomegalovirus may be detected by viral culture on polymerase chain reaction of infected blood, urine, saliva, cervical secretion, or in breast milk, although diagnosis of infection in adults usually is established by serological testing. In particular, serum sample collected 3-4 weeks apart, tested in parallel for anti-cytomegalovirus IgG, are essential for the diagnosis of a primary infection, seroconversion from negative to positive, or a significant increase in the titer is evidence of a new infection. Newborn with congenital infection can present asymptomatic, 80 to 90%. About 10 of whom will have late sequelae as part of the cases sensorineural hearing defect, exceptionally mental delay. But newborn symptomatic, in 90% of cases, will develop neurosensory sequelae, mental delay, psychomotor retardation, learning disorder, autism, hypotonia, paresis, epilepsy, vision, and hearing defects. So it's important also to screen pregnant women for cytomegalovirus. The last ToRCH member is herpes simplex 1 and 2. The seroprevalence of herpes simplex type 2 infection among women in the United States is estimated at 23%, and it's higher than that recorded in West Europe and Southern Europe. About 75% of women with recurrent herpes simplex virus genital infection will experience at least one recurrence during pregnancy, and approximately 14% will have prodromal symptoms or a recurrence at the time of delivery. Neonatal herpes is a severe viral infection contracted in almost all cases through direct contact with maternal secretion at the time of delivery. The risk of transmission to the newborn from an infected mother is high, 30 to 50% in case of a primary infection contracted near the term of pregnancy. So it's important to prevent primary infection in women during the last trimester of pregnancy, avoiding exposure of the newborn to herpetic lesion during childbirth, and in case of first clinical episode of genital herpes in the third trimester of pregnancy can be useful to perform an immunological test, since the type of infection influences the delivery mode. In particular, the presence of both virus is suggestive for a genital infection. In conclusion, perinatal death and congenital defect still represent a tremendous issue in the healthcare worldwide, in particular in low-middle-income countries. The ToRCH screening during pregnancy could have a relevant impact in reducing the burden of ToRCH infections and their sequelae. ToRCH complex screenings done by some countries, but at different time of gestation, or is not done. Globalization of the screening and homogeneous guidelines are needed to achieve a higher impact on maternal and newborn health. So, ToRCH complex serological test is widely available, and then the message should be test, test, and test. Thank you for your kind attention.
Now we have learned that ToRCH tests are necessary for specific populations and clinical conditions. Many regions in the world have strong demands for this panel as per recommendations. By medical guidelines and increased awareness towards ToRCH screening, Mindray values our customers' needs and develops high-quality ToRCH assays to improve antenatal care.
Hello, everyone. I'm Danny Yang, Director of International IVD Marketing. This is an exciting day for us all to witness the launch of Mindray ToRCH panel. With this panel, we provide not only assays with high sensitivity and specificity, but also value-added services for our end users. As part of Mindray's vision, healthcare within reach, Mindray acquired HiTest, one of the most important IVD raw material suppliers in the world, to bring more high-quality clear products to our customers. By combining the best of what Mindray and HiTest have to offer, we are now well-equipped to meet the needs of our high-end users for immunoassay diagnostics, and to provide more specialized reagents and value-added services. In the near future, we will develop more new products and solutions and more medically valuable assays for our customers. Please stay tuned.
Mindray has been holding its visions of healthcare within reach by making high-quality laboratory solutions accessible to all customers. The development of our ToRCH panel is testimonial to that vision. We hope that more people will benefit from our products and solutions.
Mindray ToRCH panel.
Improve antenatal care.
Advanced technical platform.
Results is close to perfect.
De nuestros clientes, de los pacientes.
Providing additional values.
Mindray Panel ToRCH al alcance del cuidado de la salud.